Hi, this is Dr. Jabali, and in this video, we're going to focus on topic 10.1, which is about meiosis. So what I did is gather through the past IB exam papers, all the multiple choice related to just this section. So let's go ahead and go through the process. Remember to always read the question at least 2 to 3 times. And I keep repeating this because many students go ahead and rush thinking they need to finish the time. The important thing is to really read, because by the second or the third time, that's when you fully understand what the question is asking. At the same time, you're not falling in the trap. Same thing goes with the A, B, C, and D. Do not quickly answer to B without looking at C and D, because sometimes C or D might be more specific and more right. So it's very important to look at every word, that every sentence in the multiple choice question. So first question we have here is, in which situation are the alleles exchanged? So here we're talking about alleles as it means we have, let's say, A, B, and C, and we have here, let's say, little a, little b, and c. And basically what we're doing is the exchange that is happening between two chromosomes. So we might be flipping the A here and the little a here. So this is usually happening during the time of meiosis. And we know this is actually happening at the prophase. Now, more precisely, in prophase, what usually happens is that you have a homologous right here, and which you have basically two sister chromatids. These are sisters, and these are sisters, and they're usually the same. So, for example, these are the same. You can see it's the same letters, and here you have a shorter case. But again, it's the same order. It's just, the same order of the genes, but the alleles are different. Now, the crossover always will happen here between the non-sisters. That's where it happens. So let's see what the options are. During the separation of the sister chromatids, separation, it would actually mean that the sisters are here and they're starting to pull to the opposite side. That's not when the exchange is happening, so it's no. The transmission of linked genes, transmission linked genes means the two genes will go together to the next section. So that's not exchange, that means they're going together, so no. During fertilization, when the sperm eggs chromosomes pair up, no. So when they are pairing up, they still have to go through the process of starting to become visible and then having to do the crossover, so not really. Or when the chiasmata form between non-sister. So the chiasmata they're talking about is this, right? Where they're actually going to have a crossover, where these two are going to match. So in another sense, this is what the drawing would look like, a crossover of a chiasmata, and I'm going to be putting like very simple like this, is where there going to be a crossover going on between the two non-sisters. So this is a crossover that is happening here as well as this. And this part here is where we know it as the chiasmata, is a location in which you can see that the two chromosomes are touching each other. That's the chiasmata. So the right answer is D. Now it says, which gamete can result from the following crossover? So in the crossover here, you see the drawing. That means this is big A, and this is big A, and this is big B, and this is big B. On the other hand, you have little a, little b, little b. So the crossover is happening at this direction. That means B and small b are crossing. So you're probably going to have big A and big B, the same one right here for this one. The second one, you're going to have big A, but it's going to be with a small b. So it's going to be big A with a small b. And then you have the next one, which is small a going with a big B because it has been crossed. So small a with a big B. And the last one is small a with the small b. So these are the four options. So this is what we need to search. Now, as I said this, because the crossover is happening with these two. So let's see, big A, small b. Nope, it's not this one anymore. A, big A. No, it's not this one. Although actually I should check the rest of it. The small b and this might be. Oh, look, it's big A, big B, it's this one. And then we have here small a with a small b. It could be this one. And then the big A with a small b could be this one. And small a with a small b. It could be this one. Do I put this as an answer? Many students will go ahead and put it. But here's one thing that we didn't look, is the shading colors, right? These guys are a little bit more darker. So there's a flipping going on. So the B, the small b, where this is where the mistakes will be, this small b here, it's darker. But the small b here, look at it, it's not darker, it's lighter. So that means there's a mistake here. So this is where you guys could fall the trap. Many students say, oh, it's this one for sure. Well, it's not true. Why? Because the small b it's supposed to be a small b, it's a darker one, so it's supposed to be small b darker. So it doesn't work. So let's see here, A with a B makes sense. Little a, no, it does not make sense anymore because little a also has to be darker. Now this A, so this is off. A, B makes sense. Big A, yes, big A with a small b, which is dark, makes sense. Little a, which is dark, makes sense. With big B, which is lighter, makes sense. And this, so yes, so the answer is D. This is why I told you, do not, many students will say, oh, it's this one for sure. Look at the question, reevaluate the question 2 or 3 times so you don't fall in the trap. So me going fast, I would obviously put the B quickly, right? But because I did not focus on the beginning by reading the question quickly, I didn't think about the darkness and the lightness. And then eventually I would have fallen this trap. So it's important that you read the question 2 or 3 times as well as look at the A, B, C, D. This is something I show the students all the time, is that you must read the 4 options before you're falling on the trap of IB. So once again, the answer is D. Moving, what forms when 2 different chromatids of the same homologous pair cross over? So look at this, what forms when 2 different chromatids of the same homologous pair. What does that mean? That means you have here a sister chromatid and this is a sister. So when I say 2 different, that means this one right here is different from this one, but they are same homologous pair because all of this is a homologous pair. Okay, so what forms when there's a crossover? So that means somewhere these 2 are crossing over each other. So what really forms? So if I do a drawing very quickly, it would have been like this, right? Where they're crossing each other right here at this point. And what forms here is a chiasmata. So it's not a dot or centromere because the word centromere would have been here. And you can see there's no link. No. Would it be a chiasma? Yeah, chiasma is basically a singular, so it makes sense. Chromosome mutation? No. Telomere? No. So the answer is B. So chiasmata and chiasma are basically the same thing. It's just one is singular, one is plural. So moving on to the next question. Which process could cause non-disjunction if it occurred during meiosis? So during meiosis, we actually have separations in anaphase 1 as well as in anaphase 2, right? Now, what is non-disjunction? That means there's no fail to separate. Like, for example, they are in the middle here. And look at here that it's very careful. I'm going to show you the difference between anaphase 1 and 2. Is that anaphase 1, there is the homologous. That's why I need to put 4, and this is very important. So, of course, what's happening here is a separation equally. So the pairs go in the opposite direction, right? Here we have equally the sister chromatids, one going each side equally. That would have been a normal meiosis. But when we say non-disjunction, most likely there will be a mistake. So imagine these 2 go this way. So there's a mistake. Or here, these 4 go in that direction. That will be a non-disjunction. They fail to separate equally. So what process could cause non-disjunction, as I read it 3rd time here, occurred during meiosis. First says sister chromatids do not align in metaphase 1. That's not a reason. They all do align in metaphase 1 or metaphase 2, so that's not it. Homologous chromosomes do not separate in anaphase 1. That is true. Why? First, see homologous. Yes, they are homologous. And anaphase 1, yes, anaphase 1. So this could probably, but I still will read C and D. Sister chromatids do not align in metaphase 2. They do align. They always do. Homologous chromosomes do not separate in anaphase 2. See, it says homologous, and that's for 1. Anaphase 2 should have been sister chromatid, so it's not D at all. So the answer here we have is B. That's why it's very important to know that you always use homologous chromosome on the first anaphase 1 and sister chromatids on anaphase 2. So moving to the next question here, it says, which events happens in meiosis 2 but not in meiosis 1? The spindle chromatodes attached to centromeres, that happens to both. It can happen in basically doing prophase 1 as prophase 2. Crossover occurs, yes, crossing over only happens in prophase 1 only. So here says which event happens in meiosis 2 but not in meiosis 1. So many students are going to say, oh, crossover, yes, that's just prophase 1. But the question again, look what it says, which events happens in meiosis 2. So the focus is on meiosis 2, and we know the prophase 1 happens in meiosis 1, so that's not it. Sister chromatids move to the opposite pole. Possible chromosomes become shorter and thicker by Coley. That's not it. So why I chose C? because it's only in meiosis 2 that you're finally going to have just the sisters here, not the homologous, and each one is separating. Why? Because in anaphase 1, you will have the homologous separating. So the answer is C. Next, which statement is valid regarding chromatids? Sister chromatids separate during meiosis 1? No, as we said, it's homologous. Chiasmata far between non-sister chromatids? That is true, but let's continue. Crossing over is an exchange of DNA between sister? No, it's not between sister. Remember, if this was here, these are exactly the same. A with A, B with B, C and C. If you exchange, it's going to be the same thing. It has to be with a non-sister, which is little a, little b, little c. That's where the crossover happens, so it's a no. Non-sister must have the same combination of alleles? No, they don't have. See, it's big A and small a. So we only have is B. So it's important to read all the options before you go through. Next, what process occurred during both mitosis and meiosis? The formation of chiasmata? No, chiasmata is crossover, so no. Reduction division? No, that's just meiosis because it goes down to 23. The separation of chromatid? Possible, because here it happens and here it happens in meiosis too. Exchange alleles between non-sisters. Exchange means crossover, so no. So the only option we have is C. Next, the diagram below shows a pair of chromosomes immediately after pairing the parent prophase 1 of meiosis in the cell. What combination of alleles are possible in the haploid cells produced by meiosis in this cell? So once again, that was the first time read. Now again, the diagram below shows a pair of chromosomes, yes, we have here, immediately after pairing in prophase 1. So that means right after they done what? Crossover. So the crossover has eventually happened. What combination of alleles are possible in the haploids produced by the meiosis in this cell? So that means we have to create the crossover of this. So the crossover can only happen in this action. So we can have an AB, the first one. The second will be a combination of A with a small b. The third combination could be big A with this big B, and the last one would have been small a, small b. So these would have been the crossovers, and these are the original ones. So in this process, which one's impossible? We have big AB, big A with small b, A with small b, or small b. So that would have been the option here because we're talking about the crossover happening. So the answer is D. Next, and the diagram below shows chromosomes during meiosis. So here is showing 2 chromosomes. Now, just to show you here, the chromosomes have laid down. Usually we see chromosomes like this, right? Vertically. What this picture is showing you is that I have laid down it upside down like this. That's it is. They just made it horizontal instead of vertically. So this is like I'd be flipping with your mind. Now, the next thing I did is I crossed over here and I crossed over here, which is basically this area as well, the area. What is this? This is the centromere and the centromere. So it's like I have here, this is the sister chromatids. Let me see, continue here, going like this and going like this. This is sister chromatid. And here is the other sister, all right, going here. So this is showing 2 chromosomes crossing over. Now, how many chromosomes and chiasmatas are visible? Now, you can see that there's 2 chiasmatas, right? 1, 2. So chiasmata is one of these 2. And shows chromosomes during meiosis. Is asking how many chromosomes do we see here? Now, obviously, we're seeing 2 chromosomes, right? There's only 2 options, so it can only be this. So it's not 4 because this would have been 4 chromatids. This is a chromosome, this is a chromosomes. So the answer is C. And that's the kind of questions that you will get for 10.1. It's mostly about the crossover, comparing meiosis with mitosis, and talking about the chiasmata crossover. As long as you're strong on this, you will get all the questions related to topic 10.1. Thank you.